That's crazy. Okay, good. So I'll get started then. There's like 30 people here anyway, so I'll get started. Feel free to put any questions you have in the stage chat, and I'll look at it periodically. If you need something, like if I've said something wrong, or if you need like an urgent question, ping me in chat so that I can see it really quickly. Cosine, feel free to ask questions. This should be getting recorded, hopefully. It should be working. But yeah, basically, if you have any questions, put them in the revision stage chat. If I've said anything wrong, ping me and tell me that I said something wrong, because I could also be wrong. And if you have any like urgent yes, I did test it. So it is recording. I know it's recording. I'm just hoping that it doesn't mess up in this one. I did a test recording, so it should. I will. Okay. So I'll say all of it again. I will share all the presentations I used today. I'll give breaks in the middle of each pretty much subunit. I will, if can everyone please put their questions in the chat so I can see them if there's any questions. If I've said anything wrong, or if you have any urgent questions, put them in the chat and ping me so I can see it immediately. And that's about it from me. So I'll get started then. Oh, and also my accent might fluctuate a lot, which I apologize, but yeah. Okay, so 6.3, I'm not going over 6.1 and 6.2 because Lipid already did that before, and there's, I believe, recordings up if you want to check pins in the revision channel. But yeah, so I'm going to go through 6.3 till 6.6, and then 11, all of 11 except kidneys, basically. Okay, so the first is 6.3, which is defense of infectious disease. So this one is mainly about antibiotics and antibodies and how those are created. This also links to an HL topic, but I'm just going to go in sequence of SL and then HL. Okay, so first is barriers. So the first step of preventing infectious diseases, or our first defense against infectious diseases, is the physical barriers. So firstly, you need to know what a pathogen is. A pathogen is any organism, usually microorganisms or viruses, that cause any disease. So for example, any bacteria which will cause streptococcus, for example, that causes strep throat, that's a pathogen. So there's two main physical barriers from our body. The first is the skin, and the next is the mucous membrane. So these two are the primary defense against pathogens by forming a physical barrier that just prevents entry straight away. So it's not defending after it's already entered. This just prevents it from even entering your body. So the outer layers of the skin are obviously tough. It's you can't diffuse, the cells of bacteria can't diffuse through your skin. It forms a physical barrier. Sebaceous glands are glands within your skin that secrete lactic acid and fatty acids, which make the surface of your skin acidic, which prevents growth of most bacteria. So if you were to pH test your skin, we're all a little bit more on the acidic side in terms of skin. We also have mucous membranes, so soft areas of our skin are kept moist with mucus. This is in the nose, in the trachea, in the vagina, and the urethra. Obviously the vagina in the females, and then urethra in general. Mucus has lysozymes. A lysozyme is an enzyme that kills bacteria. So in the trachea or the nose, there are also the mucus and cilia work together to expel pathogens from your body. So if you have some kind of pathogen that makes its way through your nose or makes its way to your trachea, then the pathogens will get caught in the mucus, and then the cilia will push the mucus and the bacteria up and out of either your mouth or your nose, and that's either coughing or sneezing. Coughing is usually when it's in the trachea, and sneezing is usually when it's in your nasal pathway. There are also other defenses in the form of white blood cells, of which there are two types. We'll discuss both. There's phagocytes and lymphocytes. Phagocytes are for non-specific immunity, and lymphocytes are for specific immunity. Okay, so first, phagocytes. The way phagocytes work is by endocytosis. So this is again referring back to Unit 1, where you learn about endocytosis and how cells can take in any material, any substance. So pathogens are ingested by phagocytes through endocytosis. So if you look at the image at the bottom, you can see the entire process happening. But basically, the pathogens are killed inside the phagocytes through enzymes from lysosomes. So lysosome is an organelle which has lysozymes, which are the enzyme which breaks down the pathogens. Phagocytes can ingest pathogens in the blood. They can also squeeze through the walls of blood capillaries. So if you know that, this is recalling back to 6.2, blood capillaries are only one red blood cell thick. So phagocytes are intentionally small enough or are flexible enough to be able to squeeze through blood capillaries and move through tissues to sites of infection. Because oftentimes sites of infection will be very localized, and the closest vessel would be capillaries and not any big vessels like veins or arteries. So phagocytes need to be able to squeeze through blood capillaries to be able to reach the tissues or sites of infection. Large numbers of phagocytes at the site of site of infection will be the thing that forms pus. So if you have infections like external infections, oftentimes pus will form. That pus is because of the large number of phagocytes. Phagocytes give us what I called non-specific immunity to diseases, because phagocytes cannot distinguish between pathogens. They're just there as a first mode of action to ingest any pathogen if they're stimulated to do so. So phagocytes can ingest any pathogen. It's not any specific pathogen. Lymphocytes, on the other hand, will create antibodies which are pathogen specific. Before we go into lymphocytes and antibodies and antibiotics, we have to first discuss blood clotting. So blood clotting occurs due to platelets or platelets, however you call them, are small cell fragments that circulate with red and white blood cells in your blood plasma that have an important role in clotting. So platelets, red blood cells, and white blood cells are all part of your blood plasma. Platelets actually make a large part of your blood plasma, and they have a really important role in clotting. So when the skin ruptures, because of the rupture of the skin, the damaged tissue cells or the platelets near the vessel that has been damaged will release clotting factors, which leads to a cascade of reactions that we look at. So there will obviously be reactions that are initiated by these clotting factors, where the way the metabolic pathway works, if you want to think about it in terms of metabolism, the way it works is the reactions that are initiated by these clotting factors, each reaction will create a product which will be a catalyst for the next reaction. This ensures that clotting will only happen when it's needed. So the catalyst is required to occur, so the clotting factors have to be released. One of the important products that are created through the reactions by clotting factors is prothrombin activators. So if you look at the flowchart at the bottom, it says clotting factors lead to the prothrombin converting into thrombin. the thing that leads to that happening is the prothrombin activator. So the prothrombin activator will activate prothrombin into thrombin, which is then later used to activate fibrinogen to fibrin. Fibrin is what forms the mesh of fibers across wounds and prevent further blood from being excreted or expelled from your body. So fibrin is what will create the cover that will prevent more blood from being released. But for fibrin to appear, fibrinogen needs to be altered and binded to other polypeptides. Fibrinogen is a soluble protein which gets broken down by removing certain sections of the protein structure and forcing it to bind with other peptides or polypeptides and forming a long protein fiber called fibrin. So the reaction from fibrinogen to fibrin has to be catalyzed by thrombin. That's why the entire process is really important, and the aspect of catalyzed reactions being the catalyst for each reaction being the product of the previous allows it so that that cascade of reactions only occurs if the first reaction happens. So obviously clotting factors will only be released if there is a damaged tissue or platelet, and then only if clotting factors are released, the rest of the reactions will take place, and then therefore blood clotting will occur. Okay, so the next is blood clots in coronary arteries. So this is where this is the problematic part of coronary arteries. So if deposits of plaque in coronary arteries, because there can be deposits of plaque in the coronary arteries, if those rupture, blood clots will form, which is also known as coronary thrombosis, which may completely block the artery. So if that blood clot forms, it can basically just cause the entire artery to be blocked. And then if this occurs in the coronary artery specifically, it stops blood from reaching one area of the cardiac muscle, which means that that area of the cardiac muscle won't receive enough oxygen, and so it will stop beating in a coordinated way. So you can imagine a heart beating unevenly, where there's not enough oxygen, so there's not enough energy to be able to continue the cardiac compressions or contractions and relaxations, and the entire process can't happen because there's not enough oxygen that's reaching. This is known as a heart attack. So when people have heart attacks, this is usually what happens. Uncoordinated cardiac contraction of cardiac muscle is called fibrillation, and sometimes the heart can recover after a heart attack, but if there are multiple heart attacks, they can be fatal, as contractions of the heart will stop completely. So oftentimes you'll hear that people who have heart attacks have blockages in multiple arteries. That can oftentimes lead to death, because there multiple sections of the heart will stop, and then the contractions will eventually just stop completely, and then obviously no blood circulation, and that will lead to a whole cascade of problems in terms of breathing and respiration. Antibodies and antigens. So this is where lymphocytes are important. Antibodies are made by lymphocytes, which is one of the two main types of white blood cell. Antigens are foreign substances that stimulate the production of antibodies. So antibodies are created by the lymphocytes, which is obviously there's lymphocytes and phagocytes. Lymphocytes are what create antibodies, and antigens are the substances, the foreign substances that will stimulate the lymphocyte to create antibodies. After the stimulation, each lymphocyte can only make one type of antibody, putting it into its cell membrane with the antigen combining site projecting outwards. So the way antibodies work is, did I insert an image here? No. Okay. So basically, if you, where's the image? I will insert this in here now. Give me a second. So if you look here, the bottom gray part is the lining of a lymphocyte, and an antibody. Sorry, this is an antibody. So all the Y-shaped, if you will, or cup-shaped projections from the wall of this antibody are the antigen combining sites, and obviously the thing at the top, the circle, is the pathogen or the antigen itself. So each lymphocyte makes only one type of antibody, which makes it very specific, and this is why our body has many different types of lymphocytes. And by putting the antibodies onto its cell membrane, it basically allows the entire process of antigen, causing the antigen to break down to take place. When a pathogen enters the body, its antigens will bind to the antibodies, as you can see here, in the cell surface membrane of one type of lymphocyte. So since every lymphocyte is specific to one type of antibody, only one type of lymphocyte will be able to trap the antigen. So after it enters the body, its antigen can bind to the antibodies, and after they bind, the lymphocyte will then become active, and it'll divide to produce plasma cells. So it'll create clones of itself, and together they become plasma cells, which then produce large amounts of the same antibody. Since it's coming from the same cell, they know the antibody and they know which will work for the antigen, and they'll produce large amount of the antibody, which will then bind to the antigens on the surface and stimulate the destruction. So if I go through the structure of this, at the bottom, the dark gray part and the dotted part is all the lymphocyte, and the projections are the antigen binding sites that are created by the antibody. And then the dark gray cell at the top, which is the pathogen, has projections, which are straight circles in this case. Those projections are what the antigens are. So each of those projection is an antigen, and that antigen will bind to the antigen binding site of the antibody. Is there any questions? Because I know I went through this really quickly. I'm going to scroll up just to see if there's any questions here. Now, if the MCQ paper say what the pH of my skin is, I'll know. Yeah. It's more slightly acidic. It's not very acidic, but slightly acidic. Whenever there's a question about blood clotting, the release of blood clotting factors are never the correct answer. Oh, damn. I'll look at the question. Okay. What happens during blood clotting? I'm gonna share my screen so people can see what I'm looking at. Because... Okay. During blood clotting, a chain reaction causes platelets to release antibodies to avoid infections. This is not true. Thrombin is converted into fibrinogen after. This is also not true. Sealing of wounds by fibrinogen makes platelets release antibodies for clotting. Also not true. Yeah, chemical reactions cause wounds to seal after platelets release clotting factors. This should be D, no? Is it D? Yeah? Okay, yeah. So it is D. I'm gonna assume someone's answering A. Can you point to the antibody in what case? Okay, yeah, sure. Here, hold on. Okay, so this is the lymphocyte. This area, entire thing, is the lymphocyte. These projections are the antibodies, antigen binding sites. These circles, these circles are each antigens, and this entire thing is the pathogen itself. I am using a MacBook Pro. Our school required a MacBook Pro, which is its own thing. Anyway. Okay, let me just share my screen again. Yeah, they've required a MacBook. It's fine. It's a whole thing. What's the difference between lymphocytes and B cells? I will explain that when I explain Unit 11. But if you're SL, you don't need to know. You just need to know lymphocytes. We're not bringing back chat. Stop, guys. That was yesterday. Okay. The next is HIV and the immune system. There's not a lot in this unit, so I'm just going to go through this and then I'll give you guys like a 10-minute break for any questions and I'll probably give like a question or a challenge or something like that, just because everyone hates those apparently. Okay, so HIV and the immune system. HIV is also, the full form is human immunodeficiency virus. HIV infects a type of lymphocyte that plays a vital role in antibody production. So in doing this, it basically, in a person that has HIV, these lymphocytes are destroyed over a long period of time, and without these lymphocytes, antibodies can't be produced. So when this happens, after a certain point where a lot of the lymphocytes are destroyed, at that point the condition is called AIDS, which is also acquired immunodeficiency syndrome, which is the point at which the body can't create any more antibodies and the person is susceptible to many infections which cannot be treated. So AIDS often leads to death because of the infections that occur and because the body is unable to treat these infections. HIV transmission involves the transfer of bodily fluids, as it's not easily communicable through skin contact or just through airborne, or just any normal way of communicable diseases is not possible. It has to be through bodily fluids. So the first one is through small cuts or tears in the vagina, penis, mouth, or intestine during vaginal, anal, or oral sex. So obviously any kind of sexual activity and the exchange of bodily fluids can lead to HIV transmission, which is why HIV is often considered a sexually transmitted infection, STI. In traces of blood or hypodermic needles shared by intravenous drug abusers. So if people do drugs like heroin and other intravenous drugs, and there's not proper sanitation, and there are traces of blood from one person, and then the same syringe is used by another person, if that occurs and the first person had HIV, then it's very easily communicable to the second person. The third is across the placenta from the mother to a baby. So if a mother has HIV and gets pregnant, it's a really big risk factor for the baby because the baby can very easily have HIV. Or the baby can also get HIV through cuts during childbirth or through the milk during breastfeeding. This is why oftentimes when people are diagnosed with HIV, they are less likely to want to have children because HIV transmission risk is a major risk in them. Um, the next is in transfused blood or with blood products such as factor 8, which is used to treat hemophiliacs. So any blood product that is used to treat any condition that can cause HIV transmission, or when you have to get a blood transfusion in the case where you lose a lot of blood and the blood that was given to you was infected with HIV, then you can very easily get HIV. Next is antibiotics. So antibiotics are chemicals that are produced by microorganisms to kill or control the growth of other organisms. So, for example, penicillin, the fungus is what produces penicillin and kills the bacteria. Penicillin, obviously, we use it we use it for most antibiotic treatments, but penicillium is a fungus that creates penicillin. Antibiotics basically block prokaryotic processes and not eukaryotic ones due to differences between the cells, and therefore it makes it safe for us to be able to use antibiotics. Viruses, on the other hand, don't have a metabolic pathway and they rely on host cells, so there's no way to block their processes using an antibiotic or any type of anti-metabolic drug without also harming the human cells, because viruses obviously attack the human cells itself. So for this reason, viral diseases are not treated with antibiotics. They're instead treated with antivirals, but we don't need to know about antivirals in this context. Most bacterial diseases can be treated successfully with antibiotics, but at this point, some strains of bacteria have acquired genes that cause resistance to antibiotics, and some strains now have multiple resistance, which means that if you get certain strains of bacteria, there's a high possibility that they would be resistant to the most common antibiotic, but they could also be resistant to multiple types of antibiotics. Okay, this is the last one, penicillin. So this is just, I think, just a knowledge point that you need to know. They can ask that in the, who discovered penicillin or something like that. So penicillin was developed by Florey and Chain. Their experiment was on 8 infected mice where they treated 4 of them, but didn't treat the other 4 as a control. And when the mice were infected with a deathly variation of pneumonia, and the 4 mice that were treated with penicillin recovered while the other 4 died. So initially they could only find small quantities of relatively impure penicillin, and they tested these on a man that was close to death from a bacterial infection, and he had started to recover, but the antibiotic ran out. And then after this happened, they were able to acquire more penicillin, and then 5 patients were tested, all of whom were eventually cured. Now, this process is unconventional in today's terms because we would not be able to do clinical trials that easily because of all the ethical regulations and all the ethical problems that come up with that. But this is the way penicillin was introduced. Okay, I'm gonna take a break there, and then if people have any questions, I'll look at that, and then yeah, there's basically a 10-minute break before we keep going.